my mic is on. There we go. Good morning, everybody. We'll let the kind of room clear a little bit. Thank you for coming to the Industrialization of ATMPS, Advancing Manufacturing Through Automation, Robotics, AI, and Digital session this morning. We got the coveted second to last slot before the end of the conference. We had to really fight hard to get this slot, so thank you guys for joining us. Thank you, Tim. Thank you. We couldn't do it without Tim Hunt. Thank you. Thank you very much. I'm Jason Foster. I'm the CEO of Oribiotech. Ori is a cell therapy manufacturing technology pioneer, and we're helping, hopefully, all of you bring your incredible therapies to patients at scale. I'm going to do my best to facilitate this expert group in discussing this topic, because I think of all the topics we've been discussing this week, how to bring products to patients, how to create commercial successes out of the innovations that this industry is producing, is amongst the most important topic that we could be discussing. So I'm excited to have that discussion with these experts today. I'm just going to do a little bit of a setup, and then I'm going to ask our panelists to introduce themselves. For those of you that were sitting in on Jessica's panel a few minutes ago, she made this point, and I think it's worth making again, to say, generally talk about cell and gene therapies as a singular entity. We realize that potentially the challenges facing cell therapy might be slightly different or substantially different than those facing gene therapy. So I think for the most part, we'll be mostly talking about cell therapy, probably mostly autologous cell therapy today, but would be very interested in bringing in other thoughts as it relates to automation, AI, digital, and the ability to support gene therapy or allogeneic therapies or others. So I'll put forward a hypothesis to start us out to say, in cell therapy in particular, I would say we don't have a biology problem. We've got an economic problem facing the industry. We've seen challenges from the likes of pioneers like Bluebird, who had an incredible approval success rate, bringing 3 approved products to market, but yet has been recently purchased, or at least offered to be purchased by Carlyle for substantially less than the company was once worth. We've seen pioneers like Adaptimmune, one of the first solid tumor cell therapy companies, have a tough week a couple of weeks ago as they are struggling to bring their therapy to patients at scale. So my hypothesis is that the economic challenge we're facing is one that potentially can be solved through innovation, and innovations like automation, robotics, digital, and AI. And so we're going to invite our panel to postulate on that and either prove or disprove that hypothesis during the next hour discussion. I'd like to start, if I can, by asking our panelists to introduce themselves, and I'll start to my left. David, if you don't mind starting.
Yes, thank you for the introduction. I'm David Del Bourgo. I'm the CEO and co-founder of WhiteLab Genomics. There is an S at the end. WhiteLab Genomics, we're born as an AI company in 2019, before AI was much more in the trend.
Before AI was cool.
So we've been doing that for the past 6 years, dedicated to genomic medicine, gene and cell therapy. And basically what we do, we use AI-driven approach for receptor discovery, and we have a rational guided approach to design vectors, viral and non-viral vectors. But we're also, over the course of the years, we apply our AI to design vectors that are more viable and better to manufacture. And we also worked on some projects in biomanufacturing for lentiviruses, more on the bioprocessing side, where we apply, again, like AI tools such as LLMs to analyze multiple parameters and optimize bioprocesses. We're a startup company, 40 people between Paris and Cambridge, Massachusetts, at the crossroad again of AI and biology. We're in silico mostly, and yeah, this is us.
Thanks,
Thank
David.
you.
Matt?
Matt Hewitt, I'm the Chief Technical Officer for the Manufacturing Business Division at Charles River Laboratories. I think a lot of people know Charles River for our work in research model development in animals, as well as doing GLP safety toxicology studies, but we also do quite a bit now in the manufacturing space in advanced therapeutics, and DNA plasmid, viral vector, and cell therapy, clinical through commercial. That's coupled with our biological testing business that has also got a significant presence in advanced therapeutics. And then recently we brought in our microbial sterility solutions business under our global manufacturing division, and there we do a lot of endotoxin sterility, microbial ID for a lot of the different therapeutics and advanced in cell and gene, biologics, and all different modalities, including consumer care.
Thanks, Matt. Ren?
My name is Zheng. I lead Landmark Bio, a cell and gene therapy manufacturing company based in Boston area. We are a relatively young company, about 4 or 5 years old, and we were born out of a consortium of multi-sector partnership from academia and the industry. In the past few years, we have built capabilities in cell therapy, gene therapy, messenger RNA, lipid nanoparticle manufacturing capabilities, and we have partnered with multiple therapeutic developers and help them to translate their bleeding edge research into therapeutic products. And as of yesterday, we successfully dosed the first patient. So very exciting time for Landmark Bio, and really pleased to be in this panel of experts to talk about what the future may look like for cell and gene therapy space.
Congratulations on the milestone. That's fantastic.
Thank you.
Ricardo, please. Handheld?
I can speak a bit louder.
Let's just try the handheld again. I think he's turned it on. Just speak into it. No? Can we get another handheld, please, or even a lav he can hold up?
Anyways, good morning, everyone, and thanks. It's a pleasure to join this panel and to be part with some of my colleagues.
I think we got another.
Here we go.
Here you go.
My name is Ricardo Baptista. I'm Chief Technology Officer of SmartCella.
It's gone out again. Mike's not cooperating.
It's my fault because I should have been earlier.
I wasn't going to say it's your fault because you should have been early to get your lav mic, Ricardo. I wasn't going to say anything about that.
Thank you so much. Ricardo Baptista, Chief Technology Officer of SmartCella. SmartCella is a Swedish biotech company. We operate in 2 segments. One of the segments is delivery or medical device for targeted delivery of pretty much any payload into any organ. And the other segment is regenerative medicines, where we develop allogenic therapies from induced pluripotent stem cells. And currently we have 3 programs in our portfolio: a cardiac progenitor cell program for heart failure that we plan to go phase 1 by the end of this year, a Parkinson program of dopaminergic producing cells, progenitor dopaminergic producing cells, to be in phase 1 by end 2026, and the osteoarthritis program using MSCs transformed with mRNA for osteoarthritis. We have in-house capabilities of process development, analytical development, and GMP manufacturing. And it's a pleasure to share these discussions and be part of these discussions today.
Thank you. I think we've got the full spectrum of perspectives on the panel today: therapy developers, advanced service providers, tools providers as well. So I'm looking forward to the discussion. Let's see if we can start out with kind of the macro picture, if we will. Matt, I've got a question, if you don't mind starting off, to say, it's clear that we've got therapies that work. We don't have many, certainly not for the approved products, safety or efficacy concerns. We know these products work and they work well. But yet the sort of economic model and the commercial viability model remains elusive. Can you help us understand why that is? You know, what are the major challenges to creating a sustainable industry here?
Yeah, I mean, that's a small question.
Yeah.
Yeah, I mean, so I know a lot of us have been in this for a long time, and I remember when we were having conversations about whether we could commercialize therapies, and people were actually telling us that although we were producing data that was having profound impact on patients, that this was just not able to be commercialized. And I think we've done a really commendable job of, frankly, proving them wrong. Now we're working through different steps of this process. One, we can commercialize. Two, in other areas, we're seeing instances where certainly demand outstrips supply, and I think a lot of the data supports that, including some of the data that Jason reports on a quarterly basis about patients dosed. I think there are other pockets where we do see some commercial headwinds, and there, I think one of the things that we've focused on in some of the conversations internally and externally is we need to start looking at how do we better either choose indications that have more commercial potential, because ultimately that's what's going to dictate a large degree about how successful we are, or we have to continue to work on the value proposition that we're offering to patients. Because patients that have other options for therapy, you're going to have to make a different pitch to them, more or less, to take an advanced therapeutic than ones that don't. And to put it bluntly, I mean, a lot of times when there are different standards of care for indications, then they have to make a conscious decision to go out and take something that's maybe less proven. In the case of gene therapy, sometimes they're limited, certainly on the redosing side, whereas in a lot of the cell therapy space where we're dealing in oncology, the value prop is take this or you die. And so you might think about that for a few minutes, but you're extremely likely then to sign up and take that therapy. So we need to start thinking about these types of things before we go in and commercialize, because otherwise what happens is we really start to fall in love with the science, which is the first step, but we don't stop and think, is this something that might be commercially viable? Couple that with the CMC stuff that I think we're making extremely good progress on, but we need to get better at doing out of the gate so that we're not trying to take years and untold amounts of money to get something to a business viable state on the CMC side. And I think there's a lot of room to do this for the therapeutic developers, but obviously within the CDMOs, we see a very large cross-section of the therapeutics that come through this space. And so we need to work collectively to figure out what's the best fit-for-purpose solution for these therapies.
The panel before us had suggested that maybe kind of autoimmune is kind of the killer app that maybe brings cell therapy to the mainstream, and highlighted some of the other challenges associated with much larger patient populations that are often in the community that have therapeutic alternatives. Many of those programs are early in their development still, and you're just entering the clinic or reading out some early data. Ren, if I can come to you to say, you obviously deal with lots of early programs in your company. What are some of the either product or process design decisions that you see coming out of academia or coming to your team that could be rate limiting if they try and move forward? I think we've seen the industry initially trying to commercialize essentially lab scale processes and largely being unsuccessful at creating commercially viable products.
Yeah, great question, Jason. We have seen a lot of different processes, different type of products coming to our facility, and really brilliant science. But CMC often is afterthought, and that's not uncommon. And I also think the developers are facing a dilemma. On one hand, they really want to test out their ideas in human as quick as possible to generate data, become the next inflection point to raise more money, and that's the reality we're facing. But on the other hand, we have also seen poorly designed manufacturing processes that is not reproducible, had a lot of manufacturing failures, and then raised the question about the quality of data generated from academic, phase 1, first in human trial. So it's a delicate balance there. What we always talk to our partners is that start thinking about CMC, start thinking about, imagine, this product going into patients and how that should look like, and incorporate some of the basic foundational GMP elements into the early, even laboratory protocol design thinking over there. So I think that would help, I would say, remove some of the potential pitfalls that many have experienced in the future.
Yeah, I think that we've been talking as an industry a lot about elevating this, focus on CMC and commercial viability up alongside safety and efficacy, which have been our North Stars as an industry for decades. Because ultimately, bringing a product into the clinic that's safe and efficacious, but can't reach patients, isn't really the point of what we're trying to do. And I think that ultimately has been the
I might be a little bit, take a different point on the autoimmune in that there's whether we can disagree on whether or not it's a good standard of care or not, but there's standard of care for autoimmune right now, except in the most extreme cases. So this comes back to what's our value proposition to these patients. Because the value proposition is I'll give you a cell therapy, but you got to go through chemo to precondition. That's going to be a tough sell. I mean, it is. So we need to think about, if we're going to do autoimmune, what provides the most value to patient? Is it doing the current transplant model that we're doing in cell therapy? Or are we going to move this to outpatient, potentially without preconditioning, and still see good results? If that's the case, then then we're going to have a different conversation. But if we're going to follow the same thing that we're doing now with CAR T and oncology, it's a vastly different population we'll be dealing with.
Yeah.
And separate from the CMC, which I agree with, that I think we can solve the CMC, and I'm not saying that's simple, but I think ultimately it comes down to, will the patients adopt it? And we need to start thinking about that, because we've gotten to the point where I think we've shown we can do CMC, and we got new technologies that are being developed to automate both process and scale. So now we need to take the next step and think about, what does this look like and what makes it commercially viable? Because if we address a very small percentage of the autoimmune population, then people will walk away from that and say, okay, this was a failure, and we can't do that. And I think we've had some bumps in the road recently on the gene therapy side, where we've seen very limited success because we haven't been able to really communicate to these patients why they should take this therapy.
So Matt, I think you have a really, really good point over there. It reminds me a book that I read many years ago. It's a book called Competing Against the Luck, The Story of Innovation and the Customer Choice by Clayton Christensen. It's a really good book, and it kind of take the innovation and look at the innovation from the other angle. I think as a sector and as an industry and as an innovator, sometimes we have this hammer and the nail problem. We have a brilliant technology, a shiny hammer, and we try to find the nails to apply the technology. So that is probably not necessarily the best way to look at it. And in that book, Clayton basically described, the innovation. The core idea is it's a job to be done. People don't buy products or services. They hire them to get the job done. And we really need to start thinking about, what the job looks like and what problem we try to solve. And if we start from there, we might have a better chance to have a right value proposition, identify right patient population, and apply right technologies to the disease area that is or can be solved by that technology in the most economical way.
Yeah. Thank you, Ren, for bringing innovation into the conversation, because I think ultimately innovation is going to be required on the scientific side, on the automation side, on the AI side to make this, to kind of get over the hurdle here. And The Innovator's Dilemma that Clay Christensen's famous for writing, it speaks a lot to these kinds of challenges. So let me sort of open the floor for some of the other innovators on this stage to discuss what they're doing in thinking about these challenges. So Ricardo, if you don't mind, I know at SmartCella you guys are applying advanced, let's call them technologies broadly, to try and front run some of these issues. If you don't mind just elaborating on that a bit.
Sure. I think, and I would like to give a step back regarding pluripotent stem cell therapies. It's still a young field, and there's a lot of challenges in knowing the biology, actually, of the differentiation of these cell types. So I think there's a commitment here, or a duality between bringing biology and engineering together, so that we increase efficiency of the differentiation processes, but also the volumetric productivity of these processes. So if we scale up a process that is not efficient, what's the point of scaling it up? You'll have the same issues, maybe even larger. And one of the challenges with this type of therapies is the initial processes. So we're trying to reproduce the development of an embryo in a dish. So only that is challenging. But the manufacturing costs are quite high due to the processes being, the nature of the processes. So they're still open, they're very manual. So you require grade B rooms, so the processes are not closed. Maybe some unit operations are closed, but overall the cost of manufacturing of a facility is by default very high. So what we're doing at ProCella, we're trying to combine and understand better the biology of the differentiation using analytical tools that are actually used in other industries, that you can start to understand the behavior of these cells and the microenvironment of these cells, but also what the cells are secreting, what the cells into the media, that you can understand, that you can have indirect measurements of the quality of your process and the successful of your batch, so that you can in the future predict the efficiency of the process. But we're doing still this in a dish. So we're trying to create a reference so that when we move to other culture systems, cells will behave completely different in suspension culture, but at least we have those references that we can then compare with and then do exciting process development work. But currently we are trying to understand better the differentiation processes and the biology to start increase the efficiency of the process and the volumetric productivity. That's one of our focus as well.
And doing it beforehand, you know,
We're doing it at the
prospectively.
same, so we have the privilege also of doing that, which is we want to go to phase 1 fast, and that will only be achieved with open manual processes, but at the same time we have the privilege of having technologies in-house so that we can start already developing processes in suspension culture or a culture system that is suitable for the scale that you envision for later stage manufacturing, so that you start.
Can I interrupt just 1 second? Because I heard a similar argument in a session I was in on Monday. I think there's from my perspective, we're creating a false distinction or a false dichotomy around automation, and technology is for scale, and flasks and bags are for rapid development in the lab. There's this perception that if I utilize technology early in my process, that somehow it's slower, or it's more expensive, or it's more risky. I actually argue the other way around, to say not utilizing those technologies early in the process is the most risky, because you're essentially designing to a dead end. You end up in the clinic with a subscale process on lab scale tools that we ultimately are unable to get to the point we want to get it to. And I think traditionally that has been true. The automation's been inflexible, it hasn't really been fit for purpose, and we've sort of felt constrained by it. But with a new generation that Ori's developing and others, you get the best of both worlds. You get the best biology, and you get the benefits of automation. That it's almost like buying an insurance policy. So you know if you can get the biology to work, you can smoothly transition into the clinic and to commercial scale. So I think it's unhelpful for us to think, I believe, in the old paradigm of it's either inflexible, kind of expensive automation, or it's lab tools that we can sort of use very cheaply and efficiently.
I completely agree in one way, but in another way I disagree, because we want to get to the clinic as soon as possible, and especially in the IPS, in the PSC field, clinical data is critical at this stage to understand, okay, will these therapies be efficient or not? And the processes that we take from research labs are the ones that we have to work at the moment. So use those to bring to phase 1 and make sure that they are GMP compliant, and start changing critical materials early on, but also start doing comparability studies with the process or the culture system that you envision to be used at late stage manufacturing, so that you create that bridge, that comparability bridge early on. So you can transition from phase 1 to phase 2 and already change your process, knowing that the CQA, the product quality, is the same.
Yeah, I don't disagree that that's the approach. I would argue that actually making the decision earlier gets you into the clinic faster. We can have a discussion about that.
And I hope you do that, but I've seen this story I don't know how many times. And I can tell you 90% of the time you don't switch.
That's right.
And if you look at what you have to do post-commercial, it just gets more and more complex. And then the amount of money you spend post-commercial to get to where you could have been at the beginning is substantial. And you know, we've seen that happen in the field, and some of these trials are still ongoing to do some of the process changes that they probably wish they had done in phase 1 or pre-phase 1. So I do hope you do it, but I'm just saying that the track record so far is not that on the majority.
One thing that we also do, and I think people are doing it more and more, is to first do a health economic and market access analysis early on, understand, okay, what will be the patient population and the number of patients that are expected in phase 3 and commercial. And knowing that, design, with the current manufacturing technologies available today, design how you see the process, a commercial process today for that specific needs in terms of patient population. And understand, okay, will this be viable or not? Mm-hmm. And if yes, so start working on that early on, together with the manual process that brings you up to phase 1, because to be honest, investors, they push us to, we want to see clinical data as soon as possible. We know that the non-clinical is amazing, but we want to see that in patients.
This is the crux of the issue, and I'm glad you brought it up. The biotech playbook that's for the last 2 decades has been rush into the clinic, get a signal, get us some data, then we can partner the asset, then we can fund the company. Where's that money today? We're showing them great clinical data. We're showing them great safety and efficacy. Where are those investors today? Those investors aren't investing, and they're not investing because we can't demonstrate that these products, when they get commercial, are going to deliver an ROI. So I don't at all disagree that you're being pushed by your boards and your investors to get into the clinic as fast as possible and get a signal. But that signal, that biological proof point, isn't good enough today to get funding, to get a partner. You know, we had 3 companies get approvals last year, all going to market alone. Autolus, Iovance, Adaptimmune, all unpartnered. That's an extremely unusual outcome for approved products that are clearly safe and effective. So I would argue that the biotech playbook that we've been using for the last 2 decades isn't working. I have many CEOs tell me the exact same thing. My board, I have no time to focus on commercial viability, manufacturability, to solve the problem right now. I have to rush into the clinic. But what takes time and what kills companies is not having that plan in place and having a scalable process. Ultimately, right now in this market, and unfortunately for the last 3 or 4 years, it's been that way. I want to come to David, and we'll continue to debate this over the next half hour, so don't worry, we're not going to leave that topic behind. But David, how are we using innovation, advanced technologies on the upstream production side, vector, plasmids, other to really kind of hopefully streamline production, lower cost of goods, some of those other elements that help address this commercial viability question?
Yeah. Well, I think it's still new. I guess we've seen, this emergence of digitalization and like data-driven decision-making in the upstream process quite recently. And I think there are, still need to get more accepted. And also I want to get to your point, I also agree, I mean, from our standpoint, or from my point of view, is that if you bring more innovation or more digitalization and AI-driven decision-making upstream in the process, before you get close to the clinic, I think you're going to avoid problems afterwards to push your, drug into the clinics and go through phases. At least that's also what we're trying to do when we work on designing better vectors or improving manufacturability. I guess the way to do it are, I would say, essentially to include parameters using like, AI analysis of multiple data sets, using, for example, multi-omics data, and what molecular component you can insert into your vector, what elements you can insert into like a non-viral vector to improve manufacturability, so that will be reflected eventually into the bioproduction process in the USP part. So we have actually algorithms that are predicting viability of AAV capsids that will be presented at SGCT next month. And that will be reflected in the bioproduction of like AAV. We also have projects, we work with companies on optimization of cell line engineering using AI for AAV bioproduction, for AAV production. So again, like how you use those molecular components and you optimize them. And I mean, I think it's like how to basically better engineer the process. And there is so much data, and we're talking, about, like the robotics and automatization. There's a lot of data already available. There is a lot of data that exists that's hard to grasp because it's very heterogeneous. It's, it's not very structured. It's in many places. But already with the data that's available, you can create models that will potentially improve the manufacturability of a vector, viral or non-viral, or improve the design of payloads that's going to have like the right, promoter, so you can, have the right expressions. And so I think we are in a time where the quantity of data that's been generated over the past, I would say 20 years, is just enormous. And of course, we're an AI company, so we're going to say that, but I think without AI, you cannot make sense of it. And if you want to best use it, you need to have, advanced computational approaches upstream in the process of development to impact, non-clinical phases, preclinical phases, and then have better candidates, basically, when you reach phase 1.
I want to jump into the AI ML topic in a second. I know that a new and exciting technology like that can get us all very excited, but seem a bit out in the distance. And I want to know kind of what's real today. And you've just spoken to a couple examples, which I think are great to come back to. I think Kristen on the earlier panel said, what exactly is it that AI is doing to her husband? What exactly is it that you do with that tool? I mean, ultimately, these are multivariate analytical tools. These are intended to improve processes, speed up things. So we can talk about the value that they're providing today and the potential value that they can provide in the future. I want to just land on another topic that we skipped over around the automation kind of in robotics discussion. So we've seen some new automation platforms come to market over the last few years, including ours. We've seen some new robotic solutions. Those two things often get lumped together. It's kind of all under the big umbrella of automation, and I'm not sure we all mean the same thing when we use the word. Maybe, Matt, if I can come to you and say there's a role for unit operation automation, efficiency, labor reduction, cost reduction. There's probably a role for robotics at some point in the development process, certainly, to get another percentage points out of the cost of goods and labor reduction piece. What are the sort of key roles that you see for, let's just call them automation technologies and robotics distinctly, that you're seeing applied today to manufacturing?
Yeah, I mean, we kind of separate this into two different problems. Grossly, one is, automation of process, which we're dealing with at the moment. And it's something that Ori, Lanza, Milteni, and others are certainly working to address and solve. And then there's obviously the question about how you automate most efficiently, whether it's via all-in-one or via some modular solutions, and that's a whole other debate. But I think the top line is we're deciding how to best automate the process, and then, assuming we're in a situation like we are now in some of the cell therapy space where we're supply constrained, then you have to appropriately scale that manufacturing to meet demand. And so now we're starting to potentially see some signals that there is a new and separate problem that we need to deal with, which is if you take 500 Ori Eros and put them in a suite, yes, the process is automated, but now you need a similar number of people just to manage all of those Eros. And so now we run into a different issue, which is how do we best automate the scaling of the manufacturing? And there are several companies, including here, that have presented on their vision on how we could potentially automate the scaling of the manufacturing. Certainly Solaris did a presentation earlier in the meeting. Cellular Origins is here. We've also worked with Multiply Labs. We were part of that consortium several years ago. And that's using, whether it's linear robotics or autonomous robotics, I think they both offer different fit-for-purpose use cases. It's addressing some of the same issues that if we're going to actually make tens of thousands or hundreds of thousands of doses a year, which is itself yet another debate about how you would do that, whether it's some form of centralized, some form of distributed or decentralized. It doesn't get around the centralized issue that we are going to deal with two different problems that are themselves independent, one being automation of process and the second automation of scale. Obviously, with both of those, we're doing some advanced manufacturing, and I think we've got the right people up here to talk about the AI and digitization that's going to be required to do that efficiently, because it feeds back to the central question about how do we get the economics to work.
Yeah. I heard a panelist earlier in the week say, you need to optimize the unit operation before you roboticize it. I don't think they use that word, but before you replace the human being. Otherwise, I think maybe Ricardo or someone just mentioned, you're sort of scaling up an inefficient process that, just adding more problems to the problems you already have, which I agree with. Ren, what do you see when you're talking to your clients about their scale ambitions? How are they thinking about this challenge?
Yeah, no, I completely agree with your first point. I think before you automate, you should really optimize the process, optimize your analytical methods. And those are the foundational elements you need to put in place. Otherwise, automation to me, to put it bluntly, is garbage in, garbage out process. And you can automate a lousy process, and you end up with many lousy processes, and that's not what you really want to do. And we have also seen profound impact, not just even in this industry. In previous industry, what we can see when you have a very productive cell line and what that could do to a CHO cell mAb manufacturing. I think versus maybe roller bottle. I used to work at Amgen, very familiar with roller bottle process, right? So I think optimizing the biological manufacturing process is the very first step. What we also have seen in the CAR-T manufacturing process, the first generation of CAR-T manufacturing process is about 14 days, and now the newer generations, they're 3 days, 5 days, 7 days. We know that the cost is proportion to the plant time. The longer that you keep your product in manufacturing floor, the more expensive it's going to be. So I think those are the important factors to consider before you apply automation and before you apply robotics into your process. And those are very expensive measures. They're effective measures, but they're very expensive. The second thing that I want to call out, even before we start thinking about robotic automation, AIs, having a foundational digital infrastructure is very important for any given manufacturers. And I think a lot of times we tend to focus on some of the later stage activities, but ignore having a foundational digital capabilities so that you can create this digital thread from your R&D to your CMC development, all the way to manufacturing, product release, and ultimately to hospital setting. That itself will streamline your entire process and potentially reduce cost, reduce errors.
Yeah, and I think now I agree wholeheartedly with the optimization point. I mean, work that we're doing with the likes of MD Anderson and others, we're optimizing and automating at the same time. So our system allows us to optimize very quickly. In 6 weeks, we optimize the process that they're using a legacy manual technology on, bringing it onto the system. So they got the benefits of both optimization and automation in the same step. And that's new. We've had to choose one or the other before now. And I think some of these new innovations are helping us get there. A point that you raised around digital, I think, is a great one to bring up. David, you mentioned the incredible amount of data that's been generated over the last, 5 to 10 years. I wonder if there's anyone in the room who's a therapy developer. Put your hand up if your process is fully digital and doesn't use paper. That's what I thought. Not a single hand in the room. So a lot of that data that's been generated is unstructured, potentially analog, unable to be benefited by advanced analytical tools like AI and others. So what's the interrelationship? If we want to take advantage of some of these advanced analytical tools like machine learning or AI, how do we convince our colleagues in the room and elsewhere of the importance of deploying a digital infrastructure early?
I had heard that resilience, they had no paper in the company, so.
Yeah, I don't know if that's exactly true, but we'll hopefully, yeah.
I think actually to answer that question, I think to get the adoption, you need to have good use cases. So I think the key to start with, to disrupt an industry that has like other habits, and that's normal, the inertia, are usually big, is to like prove that it can really help and it can really improve eventually. Mm-hmm. And so, because basically what can do like digitalization process with advanced computational technique is basically simulating something that you're going to optimize digitally before you're going to put it in the hardware or make it real. It's the same for AI-driven drug design. You're going to create like digital elements and make simulations on what could be the tropism or what the structure of the capsid, in the computer in silico before you're going to produce it. So I think what is important in the bioproduction automation process is really what's the use case, what do you want to achieve? So we have this project that we'll be presenting at SGCT, how to optimize the yield, production in LVs.
Yes.
So that's very concrete, and for that then you have the toolbox of AI tools that now are more known, like large language models that we use and graph neural network to basically map all the parameters and the data concerned, which is hard to do on paper, honestly. I mean, you have brilliant engineers and brilliant people, but it's hard to do on paper. You have so many parameters, so many variations. They have different measurement units. And so I think the key is to, I would say, start small, have like a good, well-defined use case, maybe not directly critical to your existing activity, and then build the confidence that it really helps, it accelerates the next phase. So what you said before, like you really solve, and what has been discussed, solves your troubleshoot, your process, make it optimal before you scale it. So that would be my recommendation.
I mean, I think it's a good question, because there's another question for the audience. How many people here still write letters and send them in the mail? I see one hand,
Miguel,
maybe two or
only
three.
Miguel, yeah.
But how many people majority write letters and send them in the mail? No, we all write emails now. Why has it been so much easier for us to switch over something that we do daily, like emails, but when we're talking about potentially much more important things like therapeutic development, we've been so reluctant to move to digital?
I think
the
David hit on it.
investment.
Yeah.
It's really,
But I mean,
it's
there
the
was an investment in IT. I mean, IT departments didn't exist 20 years ago.
But again,
now
the
they
level-
comprise a lot of companies' infrastructure.
I think it's a perceived barrier. It's, the digital infrastructure that exists today, e-batch record, LIMS systems, ELN systems, manufacturing automation systems, they're all there. They're all cloud native.
Yeah.
They can easily interlink through API. But it's this, I think, to David's point, it's the killer app. Like, why do I need to worry about that? I'm worried about getting my signal from my investors who are breathing down my neck. Like, why do I have to worry about that? And Ricardo, you brought up a point during the pre-call, which I'd like to come back to, around how you're using fully digital to save time and money and effort in the future. I'll tell you two use cases that we've explored at Ori. One, we had a partner who was doing comparative runs in San Francisco. Our team is in in London. We were monitoring the runs remotely because our system is cloud native. We saw some unusual readings in real time from the sensors in the bioreactor. Our hypothesis was, the system hypothesized that through its analytics that there was a contamination in the system. So they were running a parallel process with the Prodigy and the Eero head-to-head. They put split starting material into both systems. It turned out that both systems were contaminated. There was an upstream user contamination error further upstream in the process. We identified that 6 or 7 days earlier than they would have known otherwise in their existing platform, because it doesn't have that analytical capability, saving them 6 or 7 days of labor time and effort and money, 6 or 7 days worth of media, reagents, vector additions, and other things. A basic use case. We can monitor the cell culture health and status from anywhere in the world. That has tangible value. Another small kind of tip of the iceberg use case is cell count. In real time, we can tell you how many cells are in the bioreactor at any given time. If you're on day 3 or 4 of your process, have you hit your target cell yield or not? We can tell you without you having to go into the clean room, take a manual sample. These are the beginnings, the tip of the iceberg of the use cases that may hopefully convince the industry that digitizing early is a benefit. But I know, Ricardo, you're using predictive analytics. I don't know if you use the word digital twin, but you were sort of in and around digital twins.
Just to give a step back on saying that, I completely agree that you can optimize and automate at the same time, rather than first optimize and then automate. Why? Because you can optimize in a static culture, and you apply DOEs and you identify your design space, and then you change to a suspension culture and then realize, okay, I'm completely out of the design space that I just identified. I think on that note, scale down tools play a key factor on that, where you can high throughput different parameters of your culture system, and using our approach is generate as much data as you can, during early stages of development and use analytical tools that you might be able to use for monitoring or control your process, like Raman, LCMS, metabolomics, proteomics. So use those analytics to understand your process early using scale-down tools for high-throughput analysis. However, these scale-down tools don't exist for all unit operations. But at least we have something. Regarding the use of digitalization tools, we're working, so we use digital twins that in parallel with DOE approaches, you can actually optimize or have a virtual model of your process where you can simulate changes or disturbances and understand the impact of those in the process. One thing that we're doing regarding robotics, we're applying robotics early on in the manufacturing, so in the warehouse, for example. We're applying robotics on that. And also in the analytical preparation of samples. We're using robotics to standardize sample prep for QC to remove variability between operators, especially in assays such as flow cytometry that have a lot of steps, a lot of preparation steps. So we're using robotics for that. We're working with a company, a Swedish engineering company, ABB, and they have a very, very interesting toolkit of robotic arms and scale down centrifuges. So it's we're doing that in terms of robotics.
Yeah, one of the interesting things about that kind of in silico approach, digital twins, and being able to understand our process more fundamentally, but maybe before it hits animal testing, certainly before it hits the clinic. We have a reference data set of 800 runs in the system that we bounce our partners' data and process off of. And that's one of the ways in which we're able to optimize so quickly. We optimize in tech transfer, the MD Anderson process I mentioned, in 6 weeks. Doing tech transfer in 6 weeks saves a huge amount of time if you're trying to move your process forward more quickly. So I think those kinds of digital tools are underutilized and maybe underappreciated currently in the industry. We've got about 8 minutes left. I've got a crystal ball question for my panelists, a little bit about what are the breakthrough technologies that are maybe not yet widely adopted or understood that you're most excited about, that you think will have the biggest impact in the next, let's just say, 3 years time horizon? I'll let whoever wants to take that one volunteer.
I can start
with
at least for pluripotent stem cell therapies. I think, and it's not directly related to automation, but the use of synthetic biology tools are extremely powerful for forward programming, so to control the differentiation processes, to remove populations, undesired populations, and then you can actually automate the addition of molecules that will trigger these on-off switches for specific molecular cues. I think that's quite exciting, and also understand early on what I was mentioning before. Draft your manufacturing processes for commercial manufacturing to understand the cost of the manufacturing, but also the scale that you will target.
Yeah, Doug Dennison from Bayer made an interesting point on a panel earlier. We don't want to work the crowd too hard, but I'll ask you, for those of you developing therapies in the crowd, in your TPP, in your target product profile, how many have you included things like cost of goods, number of patients intended to be treated per year, intended line of therapy, number of doses per year? Anyone? Miguel, excellent. Kristen, excellent. A couple of you. Very good. These are the kinds of things that, if we're developing small molecules or biologics, we're not also often including a target cost of goods in our TPP, but this is something that I think has becoming quickly industry best practice to say that, we need to know what we're shooting at, to Ren's point earlier. We need to know what we're shooting at longer term to be able to work backwards to know if we've gotten there or not. And we don't end up at the end of the destination and realize that the bridge isn't there. And so that's great to see. In the last 5 minutes, I'm going to put each of you on the spot, if you don't mind, with a question. Ran, if I can start with you. Landmark is pioneering different models for scalable manufacturing. You've just gotten a new, I think, financial partner in your business as well. What do you feel are the kind of foundational elements that you hope to put in place for your business to help enable this next generation of therapies to reach patients at scale?
Yeah, happy to address that question. So in the past couple of years, we have partnered with some developers to really scale different type of manufacturing, including distributed manufacturing. So we have a lot of learning through this journey. For example, in order for us to scale decentralized manufacturing, reproducibility of the process and analytical methods in different facilities and labs by different operators, it's really important. And if your process and analytical method is not reproducible, and it's impossible to have more than one manufacturing size. So, to me, I'm going back to, the foundational elements. The process need to be robust. The analytical methods need to be robust. Those are really important. And another factor I think a lot of people don't always talk about is the operability of the logistics, including product release, quality oversight, and those are the things that are very important. And they are often appear to be invisible because everyone focusing on manufacturing, but those ancillary processes, and they're like the sands in your shoes. When you climb the mountains, they could really kill you. So those are the things really important. And finally, and I'm so glad that you mentioned about cost of goods in TPP. I think we as a sector, our biggest challenge is margin. We have blockbusters, and we have successful products that give patient cures, but the profitability for this industry has yet to come. And I think that's one of the biggest challenge when we think about manufacturing processes or think about given products, and having an acceptable cost structure or make the cost structure acceptable is really important.
That's right. For the sustainability of the industry, we have to be thinking about those kinds of things. Matt, how are you helping your clients integrate technology broadly, like the Eero system and others, into their manufacturing thinking, their process development, their clinical scale up? Like, how is it practically being applied by Charles River? Yeah,
I mean, I would say, I'll take a different way to the answer to that question, which is that we actually just remove the client a lot of times. And we proactively go out and look at the space and the technology and then ensure that we understand how to use all the new technology. And I think the Eero is a good example of that. That's something that we proactively did with Ori. We've done this with others, because if my teams are learning to use a technology when a client does come in for the first time, then I'm probably already behind in a lot of ways. So we do that, and I agree with Rand's point as well around how we overlook certain things, and that's another thing that we spend a lot of time on with clients. Is not just process, but QC and logistics and warehousing. And it's not really a joke, but I do kind of joke with my operations people that I know they're working because I know they're doing what they're supposed to be doing because I don't know they're working. Because if operations is going how it should, then you don't have problems to deal with. And so, but that, believe it or not, when we do a lot of this stuff with clients and potential clients, one of their primary concerns outside of process and QC and GMP is like, what is your logistics? What is your warehousing? What's your sample management look like? And what are those systems? Yeah. No, I always love it when I walk into the lab and there's no one in there, because the machines are doing the work and they're off doing other value-added activity. I think we only have time for one more comment. David, I'll leave it with you. How are you seeing the kind of AI in the loop and these kind of advanced tools accelerating process development and optimization?
Yeah, yeah. I think it's key. I think it can be accelerated. They are in the loop, like the discussion between the in silico models and the in vitro. You say you work in the lab, there is no one. It's like everything automated. I think there are still like progress to be made on designing in silico assumptions and testing them very quickly in vitro. This lab in the loop can be improved, and we're working on it to improve it in the next, in the near future.
Excellent. So in summary, I think optimize and automate early in your process. Think digital because it pays dividends if you invest very limitedly even in the beginning. And ensure that we're not falling into the same traps that we've been falling into and designing to a dead end, because patients deserve the incredible science that you guys are developing. So thank you very much to the panel.
Thank you.
Thank you.